But it's really nice to be able to have the opportunity to present what I'm doing and what I'm doing here at the CRE and to present it to such an impressive group of people. So I'm going to be presenting to you a project called SynBio for All, which is a platform that we're trying to develop in order to bring synthetic biology to the public and to the non-scientific world in general, in order to bring about public education in science and engagement as well. So I'll be using those words a lot, education and engagement. There's the original title slide. And the SynBio for All project is a pilot, as I said, of the bigger Citizen Cyber Lab project, which is an European Commission funded project under the 7th Framework Program, which was funding programs between 2007 and 2013. It's a 3-year project that is in its last 6 months, so we're wrapping everything up, trying to get all the data together for the final review and everything. And this project involves many different partners. So there are 7 partners involved in this, representing 3 different countries. The UK has representatives from Imperial College of London, University College of London, the Mobile Collective, who helps us do organize events like this and this kind of thing to get our work out there. There's also involvement from groups in Geneva, so UNITAR and the University of Geneva and CERN. And of course, the University of Paris-Descartes, representing France, and it's Paris-Descartes and the CRE who is doing SynBio for All. So all of these other groups are also doing their own platforms, putting forth their own science, and it's really interesting stuff that they're doing. So if you want to see what other kind of things are going on in the CyberLab project, you can check us out on the website there. So SynBio for All, as I said, is looking to open up the world of synthetic biology to the public by really involving them in every aspect of carrying out a scientific research project, from the understanding of the need to address a problem to coming up with ideas in order to address this problem, and then basing those ideas, using those ideas to create a project to address this, and then to implement that. And so we really want the people involved in this outside of the lab, the public in general, I mean, to be able to help us study, to give us support, to help us in the creative process, and most importantly, to learn. In order to do this, we are attempting to create a synthetic biology community. As somebody who did a master's in kind of a smaller university where we didn't have much money and you didn't have a lot of avenues to go out and reach out and ask people for help, having this kind of online community would be really an amazing thing. And it's already building kind of out there in different places. But we would like to design a platform, and we have designed a platform, we hope, which the synthetic biology community that we build will be able to interact with one another, to exchange ideas, and again, importantly, to learn. So this, in case you don't know or you're not really familiar with the ideas behind synthetic biology, this is an aspect of science that is very exciting to me because it has probably the most potential of anything out there. I think Bill Gates said in an interview with Wired magazine that if he was a kid now, he would be doing biohacking, because it has a huge amount of potential, from the making micro factories from bacteria or different organisms to produce virtually any molecule that you want to produce, to doing real drug delivery, and I mean like the most literal aspect of drug delivery you can think of, where you design a DNA molecule that you can put a drug molecule inside and lock it like a little chest, deliver it to a cancer cell, it recognizes a receptor on the cell, it opens up, delivers its molecule and kills the cancer cell. I mean, this is some really cool things that you can do with this. And the difference between synthetic biology philosophically and regular biology is where in regular biology you are looking at how things work and trying to understand what the mechanisms of life are. In synthetic biology, you're approaching more from an engineering standpoint. How can we change things? How can we take things from different systems, put them together, and create something new or create something useful? In order to do this, you have to take these. Usually we work in DNA. I'll just try throwing that out there. And what you want to do is you want to try to abstract the ideas of these parts that you can move from one place to another to create new things. So these little symbols here represent a gene cassette. Okay. And these parts are abstracted. The different parts of the gene cassette are kind of abstracted in order for people to understand it. You don't really care how it works, you just want to know what it does. So the arrow represents a promoter, the little green blob is a ribosomal binding site, the big sideways arrow without the kind of tail on it is the actual protein sequence, and then you have a termination signal at the end. And the idea is you can take different parts and mix and match them in order to create the level of protein that you want. Maybe you want less, maybe you want more. But the idea is that you can use these and exchange them with other people and this kind of thing in order to build the system that you want. So the current largest system right now for doing this kind of abstraction and for having a kind of optimized repository for these parts are called biobricks, where they're standardized, where they can be easily placed in a cell, taken out of a plasmid, moved from one to another in a system very easily. And these are done under the auspices of the IGEM competition that I'll talk about later in the talk, which is a competition for molecular biology students that takes place every year. And again, the idea behind all of this is to take biological systems and either remodel them or to create them from scratch. So some of the applications for this are in drug production. Here you see how the basic scheme for producing insulin, which was one of the first drugs to be produced through kind of a synthetic biology approach, where you have two protein chains which are coded into plasmids, which are produced in two different E. coli bacteria. Those protein chains are isolated and purified and then are joined together through a chemical bond, through a chemical step. So the first step of this would be a synthetic biology application. And this was done in 1978 and was kind of the beginning of this whole system of creating drugs this way. This is one of the newer drugs. This is an anti-malarial drug called artemisinin, which is, it's created in yeast, and it was first proposed, this kind of approach to creating artemisinin was first funded through the Bill and Melinda Gates Foundation back in 2003. And whereas previously it was produced from plants, it is now produced in yeast, and there are vast quantities of this drug produced now for very, very small amounts of money. So this is very exciting. Then you have the food industry applications. These are probably the most controversial among the general public because food is something that everybody has to eat, right? And if you don't understand what kind of the processes behind synthetic biology are, maybe you're uncomfortable with with this type of approach. It's kind of a goal of SynBio to try to help to disabuse people of what kind of false notions that they may have about GMOs and if they're bad or if they're good. Certainly there are aspects in their implementation that might be less desirable than others, but we really hope to be able to educate people about what exactly a GMO is and how exactly it affects its environment and people. There's also, as I said, the potential to create materials and to create fuels. So the biofuel industry is something that people had put a lot of hope in, and it still kind of hasn't reached the level that people had thought that it would reach by now. So if it's something that you would be interested in, it would be something that people could kind of come together and help to study. So the last point was going to be synthetic organisms, and I just want to mention so I gave away the joke already. But as I was mentioned in Samir's talk, the cost of making nucleotides, of doing DNA sequencing is just plummeting. And as it becomes easier to synthesize DNA, as it becomes cheaper, it offers a lot of opportunities to kind of redesign organisms from the very basic, from just one gene or another, to doing it on a large scale. There's already been famously in around 2008, 2009, the synthesis of Mycobacterium genome. No? Sorry. Mycoplasma genitalium, thank you. And up to now yeast, trying to create synthetic yeast cells. And I was trying to make the point, we don't know what the future could hold for creating synthetic organisms. So the platform overview for SynBio, for all the basic kind of flow chart of the approach that we want to take when helping students, is to give them the opportunity to learn, to, as I was saying, learn about a potential problem, have the ability to propose a research idea. Those ideas get coalesce into a project. If we have multiple people proposing different kinds of projects, we can have kind of a competition to see which one has the most promise, which one is the most feasible, given the resources which are available. We can then maybe translate that into the lab, where the scientists who are helping the students come up with these ideas and stuff can take the best project, bring it into the lab, try to carry it out, keep a dialogue going with the online participants or the non-scientific community in terms of what's happening, maybe exchange new ideas. Then when we get data, we analyze that data, and then we're able to disseminate the results. So we are doing this project in association with the iGEM competition. Paris Betancourt and the CRE has an iGEM team. So iGEM is the competition for internationally genetically engineered machines, which takes place every year in Boston. So over the summer, different student teams get together from around the world, get together, and they come up with a project. They have the summer to work on it, and then in the fall, we have the Jamboree in Boston, where they compete to see who has the best project. So last year's team was a very nice project. The project was called The Smell of Us. It was trying to understand what kind of smells people produce, how people perceive those smells, what they mean, how can they be controlled, can you make somebody smell nice, or if they don't maybe smell that great. And so in reaching out to the public in this, the team itself was made up of 15 researchers. 9 of them were from outside of the CRE. They organized public events at Cité des Sciences, which is this big place for science in the north of Paris. La Paillesse, which is kind of the DIY science group here in Paris, and also at Saclay. They got over 500 participants to get involved in this. And what they would do is they would do a test where they had made these cotton pads and they would put them under their arms. You dance around a little bit and get sweaty, and then you take people's name off of it, and then you give it to somebody else, and they smell it, and they tell you what they, does this person smell good? Do they smell bad? What kind of smell do you smell? And in collaboration with a team in Rennes, they were able to make a game called the Smell Game, where they could input these smells into the computer, and the computer would give kind of personalized feedback to what you actually smell like based on what other people were saying. In addition, we have a community on Facebook of over 300 people who kind of follow us and communicate with us. So from this data, another aspect of the project was to create kind of this odor library. So these kind of biological parts were made that can help people to kind of understand how smells work. So they make these different kind of molecules, from banana smell to sweat to rain, this kind of thing. And again, we were able to collect a lot of citizen data through this kind of thing. It's the kind of science that is interesting to people because it's personal. And when it's personal like that, then people can really get involved. And another aspect of the project that they used in order to reach out to the people were creating MOOCs, so massive open online courses, which I will show you an example of that in a minute. But I mention it now just to mention that it was the last year's team that started this, and that we are going to continue with adding to the MOOC. This year's project is quite interesting. It doesn't yet have a super sexy title, but this title is pretty self-explanatory. The project involves fortifying fermented foods with different vitamins through genetic modification of the microorganisms which are involved in carrying out the fermentation of these foods. So we are specifically looking at lactic acid bacteria and yeast, and we're trying to transform the team is trying to transform these microorganisms with these different vitamin pathways. And the idea is, if you have a community, say in India, we're currently targeting India, we have some Indian experts on the team. If you have a community which eats a lot of fermented foods, but perhaps they have a vitamin deficiency for one reason or another, you can give them maybe these microorganisms which will produce this vitamin. And in this case, there's no patrimony involved. You're not trying to tell them to change the way that they do things or the way that they carry out their everyday life. You just give them a thing, okay, put this in your food, and they do it in their traditional methods, and hopefully we help to address a specific problem. And in this way, this shows also the potential of synthetic biology to recognize a specific problem somewhere in the world and then quickly carry out a process to address this. So we are doing, in this project, reaching out to the people, fermentation workshops. There's a fermentation workshop with the iGEM team tomorrow. If you want to go check that out, it will be very interesting. We are making different kinds of food. So this is showing how we make idli. So this was made over in the iGEM workshop, where you just mix some rice and you mix some lentils. You leave them in a bowl, and overnight they ferment, and you have this nice batter which you can then steam and create these really tasty kinds of food, which are eaten all throughout South India. We hope to interact with the people in order to get their opinions on this, of course, by doing interviews and surveys. If you take part in the fermentation thing tomorrow, we'll ask you to do a quick interview and just a quick survey rather, to understand what maybe you think about this kind of approach. And we are also hoping to contact people in India in order to get their ideas about what they know about how their food is made and how open they would be to this kind of thing. Okay. The second point for our citizen interaction is the actual platform that I was telling you about for interacting with the public, which is called IdeaWeave, which is a software or an application that was developed in-house at the CRE, and it's Jesse Himmelstein who is doing that. And lastly, also we're continuing with making the videos and making the MOOCs. So here's what the MOOC looks like that was developed by last year's team. It was really targeted at high schoolers and getting high schooler students involved and interested in joining an iGEM team. It was done on this Genius platform, which is a website which was originally put together for posting music lyrics. But there's all kind of different categories where you can add things to. And so the team made a category for all kind of videos related to iGEM, and it's really nice. You have videos on how to create an iGEM team, videos on how to do the basic techniques involved in synthetic biology, like restriction enzyme digest, running a gel, this kind of thing. What a biobrick is, for example. They have over 20 videos uploaded to the site so far, and we're hoping that we can add some more videos this year through the iGEM team's 2015's project. In addition to creating the videos, what's nice about this site is that it has a reward system involved, where whenever you create something where you want to involve people online, because the Citizen Cyber Lab scope is about creating these digital online platforms for getting regular citizens involved, it helps to have what they call gamification, where you give people a bit of a reward for actually participating. And in this way, we have a reward system where you get kind of points if you make an article or if you edit something or this kind of thing. So that's really nice for the people involved, and it tries to make sure that they come back and keep participating. So this is IdeaWeave that I was mentioning. It was originally conceived as kind of a creativity incubator, social media platform for science students and citizen science. So the basic idea behind IdeaWeave is that you, of course, because it's social media, you're able to post things and to interact with one another, but you're able to post three different aspects: a challenge, an idea, and a project. So you would create on the website, say, a challenge where you say, okay, how can we use fermented food to address hunger or malnutrition? And then somebody else may propose a different idea for how we can address this. And then somebody else might say, well, I think that idea is a good idea, but maybe we should do it like this. And from there, you can then propose projects and this kind of thing. And then you have a real interaction among the community as far as this goes. So these challenges represent a method for crowdsourcing, a method for creating events and getting people involved. And so I'll be doing a little 10-minute presentation on IdeaWeave tomorrow that people can be using during the Think Camp. And the website is there, ideaweave.io. Okay, here's just a few more of the functionalities of it. There's project workspaces. You can have a web page. There's an Etherpad embedded into the site that you can use, and there's also Trello, where people involved on the project can have a little space where they set deadlines and interactions with each, project goals and this kind of thing with each other and try to organize stuff. There's should be a chat feature and an activity stream. They should be there. It's still kind of under construction, and but it's coming together. So just finally, we in the CyberLab project are trying to measure learning and what these platforms that we have created can help, how these things help citizens learn science and this kind of thing. So we are doing with Laura Kutzler and these kind of interviews with the iGEM participants and with the participants of the other kind of aspects of the project. And with the iGEM students in particular, we are looking at trying to understand how carrying out an iGEM project helps the students to learn how to organize things, learn the methodology behind the science, learn how to do the lab work, and then analyzing the data that they get from the lab work. And also, very importantly, how did they learn how to collaborate with people? How did they learn how to reach out to other labs and ask for expertise and ask for materials and this kind of thing? And these, again, are all kind of under the iGEM umbrella for this. Okay, so here's some ways to get a hold of us, to contact us, or just to follow what we're doing. We have a Facebook page, we have a Twitter feed, so at SynBio for All, and there have been a couple of articles written about us in the popular media. So it's kind of a long URL, but if you Google SynBio for All, you'll come across these websites, or these articles rather. So these are the people that, of course, it's not everybody that I want to thank, but you have the iGEM team members, the different advisors, so Ariel, of course, Daniel and Laura, who help us analyze the data, and then our other partner institutions. And so, thank you.
And again, if you have any questions